Good afternoon, everyone, and thank you for standing by. My name is Barbara McMakin, and I'm from the NINDS Office of Neuroscience Communications and Engagement. On behalf of the NIH, I would like to welcome you to this afternoon's call and to thank you for your interest in participating in this discussion with us today. Today's call is being recorded. If you have any objections, please disconnect at this time. Dr. Vicki Whittemore, Program Director of NINDS, will introduce the speakers, each of whom will make some remarks, after which we will answer your questions. If you have a question for our speakers, we invite you to submit it through the Q&A box at the bottom of the Zoom screen. We will try to make our remarks brief so that we can answer as many questions as possible in the time we have available to us this afternoon. I also wanted to mention that we are exploring different formats for these telebriefings going forward. For our next telebriefing, we plan to include live oral questions during the question and answer session. That telebriefing has not yet been scheduled, but once we have those details, we will send out a message to the listserv and post the call information on the MECFS website. Now I would like to hand the call over to Dr. Whittemore.
Thank you very much, Barbara, and apologies for the delay in getting started. We've been waiting for Dr. Koroshetz to join us, who I see is now here. So I will turn it over to Walter for some opening remarks.
Again, sorry to be late. Working on a COVID project, which is relevant. We'll talk about that. So I want to welcome everyone to our first telebriefing of 2022. Several developments to talk about. Dr. Joe Green from NID will let us know what's happening in the consortium. Dr. Vicki Whittemore talk about the Trans NIH MECFS working group. Dr. Nath has a conflict as well this afternoon. He may be able to join later, talk about analysis going on in the intramural study. And we're really gratified to have a wonderful guest speaker this afternoon, Dr. Benjamin Nadelson from the Neurology Department at Icahn School of Medicine at Mount Sinai, where he leads the Pain and Fatigue Study Center and has several grants from NIH. And he'll talk to us about his current and planned research studies. We're always eager to hear from you, so we'll keep our own remarks very brief before answering your questions. We did receive a number of different questions about the Recover initiative, which is actually where I was just a second ago. So it's NIH's large-scale effort to understand why some people do not fully recover from infection by the SARS-CoV-2 virus. Many people experience long-term symptoms, and a large number of these symptoms overlap with MECFS, and the expectation is that many of these people, after the 6-month period, will actually meet criteria that have been established for MECFS. So unfortunately, the COVID pandemic is looking to really worsen the problem of suffering due to MECFS. In the Recover program, we're concentrating on trying to understand this post-infectious persistence of symptoms. Of all the symptoms, fatigue is the most prominent. MECFS investigators and clinicians are involved in the research and collaborating with the Recover investigators. They're also involved in the oversight committees and working groups of the Recover initiative, of which there are many. We're going to have an upcoming Recover webinar. So this is for the Recover investigators and people who are interested in it. It's going to focus entirely on MECFS and the lessons learned there. Recover is a special situation. It's an ability to closely study a large number of people as they recover from the same virus at the same time. It's unfortunately a natural experiment that we've never been able to do in MECFS, but one of the silver linings of the horrible pandemic is that we can examine the transition from acute infection to the chronic condition, look for clues as to why some people develop long COVID and others do not. And it's likely that we will find in these individuals biological drivers that are relevant to MECFS. We're also hoping to start clinical trials in people with long COVID. And if anything works in that large homogeneous population, I think one of the first things is to try and move it and see if it generalizes into the greater MECFS population, where we don't actually know what the virus is. Some people, it's not clear it is a virus. Some people, it does seem to be a virus. very heterogeneous ME/CFS compared to COVID. And also the COVID situation is fairly new. We're looking at people who are just months or most a year or two out. There's an ME/CFS. The heterogeneity is such that, unfortunately, many people have been suffering for decades. So COVID is a much more pure way of trying to get at the bottom of these problems that ME/CFS has been trying to get at, but hopefully we'll have better success because of the advantages of this natural experiment. See, I have a couple of questions. Barbara, did you want me to continue with the questions, or will you do those later?
We'll do questions following everyone's opening remarks.
Okay, very good then. Thank you.
I guess that's me. I'm next. My name is Joe Breen. I'm a program official, and I work closely with Dr. Koroshetz and Dr. Whittemore. And I just wanted to give you a few introductory comments about the status of the collaborative research centers for ME/CFS, an NIH-funded effort. Suffice it to say that there has been vigorous activity across the 3 research centers and the Data Management and Coordinating Center. They're hard at work, really, in year 5 of their funding, and you know, there were some slowdowns due to the pandemic, of course, and I think that is now they're really working hard and at full function, and that would be reflected in some of the output, including a number of preprints that I just wanted to make you aware of as an example of the productivity that I think we'll expect to see in the next few months. The Columbia Center has a January medRxiv paper noting some peroxisomal dysfunction and dysregulation of a choline pathway in ME/CFS, which is a novel finding. The Jackson Lab Center, led by Durya Unitmatz, has a bioRxiv preprint about multi-omics of host microbiome interactions, utilizing some of their really groundbreaking and really some leadership in terms of microbiome studies that are occurring there at Jackson, in this case, applied to ME/CFS. And the Cornell Center, led by Dr. Maureen Hansen, has continued to be productive. They have a number of papers, the most recent of which is a plasma proteome study, as well as some interesting recent reviews that I would encourage folks to look at. And the Data Management and Coordinating Center has really 2 things that we've mentioned before, but I think are really going to be there are already assets to the community and the research community, and I think they'll be increasingly so. The MAP ME/CFS data management structure, which is actually, there's a preprint available on the website. If you go to MAP ME/CFS, you can find that to really see all of the powerful things that the MAP ME/CFS organization can do, and the organization of research, really, so to eventually allow cross-study comparisons and the kind of things that we weren't able to do before in this field, which I think will be a big advantage. So I don't want to spend too much time. I want to leave time for others on this, but suffice to say that there are a lot of projects that are reaching the stage where they'll be peer-reviewed and available to the public in these areas, including our Canadian collaborator, the lab of Dr. Alain Moreau, who isn't NIH funded, but works very closely with us. So I'll stop there, and I believe, Vicki, you're up next.
Yeah, thank you, Dr. Breen. I'm Vicki Whittemore, a program director at NINDS. And just to tell you what the Trans NIH ME/CFS working group has been up to, as most of you, I think, know, we released the request for applications to continue or for funding of the centers, as well as the Data Management Coordinating Center. So those were delayed a bit, and so we're working in their release. So the deadline for applications is in May, and they'll be reviewed this fall. And Dr. Breen and I are working closely with all of the investigators to make sure that they can get through that gap period in funding with either continuing their work with no cost extensions, which means using funding that they still have available to them, just extending the year of their award, or providing supplements so that the research will not come to a halt at the end of their funding period, but they'll be able to continue with their data analysis and writing papers and continuing their work. We got a question in advance from Court Johnson about the funding for these centers. And as I explained in the newsletter, that our first newsletter that we sent out recently, when Congress appropriates money to NIH, each institute receives a budget. And within that budget are funds for what we call intramural research, which is where Dr. Nath works. So
Stopwatch.
So those are the labs that are actually on the NIH campus. And then there's the extramural budget, which is research funding that goes to investigators outside of NIH, like Dr. Nadelson, like funding for the centers. And it's funding that is out of that extramural budget that is designated for the center. So it's not a mysterious place where this money comes from. It's money from that extramural budget that gets designated for those requests for applications. So it's been exciting. I've been contacted by several new groups who are interested in submitting applications. And so we'll see who competes for the 3 centers this summer, fall. And with that, then I would now like to turn it over to Dr. Benjamin Nadelson, who it's our pleasure to have with us this afternoon. I'm the program director for Dr. Nadelson's grants and have really enjoyed interacting and interfacing with him over the years. And so we'll turn it over to Benjamin to talk about his recent and ongoing research. Dr. Nadelson.
Hi, thank you very much. Do you see my slides, Vicki?
Yes, they look great. Thank you.
Great. Okay, well, first, let me thank you and Dr. Koroshetz for honoring me to update you about where we are here in Mount Sinai. I need to thank Michelle Blate, our center nurse practitioner, and Tiffany Soto, our study coordinator, for making the Pain and Study Center research limb work. And certainly I need to thank all of you, patients who have been or will be in the work that I'll review in this talk. So the first funded study I'd like to present to you has to do with our, of course, belief that CFS is a brain disease. And for me, myalgic ME is myalgic encephalopathy. And if you want to know why, you can ask me that in the question period. But our plan is to collect spinal fluid from CFS patients, I'm going to say CFS just for brevity, and to identify every protein in spinal fluid, to identify biomarkers that are specific for this disease, and this will allow a lab-based diagnosis. So if you look at the figure on the right, that's the results of our first study, which was very exciting because that red part of the figure indicates that there are 738 proteins in spinal fluid that differentiate it from patients that have fatigue after Lyme or healthy controls. But there's a problem with this early study, and that is that the technology available then required us to pool spinal fluids. So we essentially had 43 patients, and we had to pool them all to be able to do the analysis. And so the problem was that since we had to pool, it's possible that many of the results, many of the differences may have been to just a few subjects exerting those differences due to our need to pool. So then, because of that, we moved to request additional funding for a new method not requiring pooling. And there we received a funding from NINDS, and we have new set of spinal fluids from chronic fatigue and from sedentary healthy controls. We're going to get more spinal fluids from our colleagues, Dr. Dans Peterson or Avi Nath, in order to factor out proteins that are not specific to the fatigue of CFS. We're going to add samples from patients with multiple sclerosis and other neurological diseases that do not have CFS. And the team we put together to do this is, I think, fabulous. Besides me, there's Steve Schutzer, my colleague at New Jersey Medical School. Dr. Smith is the person who now has been involved in developing these new proteomic methods that let you to identify the protein in a single sample of spinal fluid, and our partner Tao Lu, who is actually working to make the analyses happen. So I'm having trouble getting the next slide. There. So our second funded study has to do with the fact that my postdoctoral fellow was identified a new way of looking at sleep. He looked at every change among sleep stages. So when you go to wake, to light sleep, and to deeper sleep, each of those is a transition. And he did this transition analysis, which found differences between patients who had CFS alone and others who had CFS with coexisting FM. And so the question that we wanted to look at in this study is, are these two illnesses the same, or are they different disease processes? Because some say they are the same, but that's just a belief. So here's the data on the right from our sleep study. And what this figure shows you is that patients with CFS alone have different sleep transition patterns than those who have CFS with FM. The CFS alone transition from REM to wake, the red arrow, whereas the patients with CFS plus FM transition from wake to deeper sleep and then are aroused, going from N3, which is deep sleep, back to lighter sleep. And then we found a paper that indicated that if you sleep on a rocking bed, it improves all that stuff on the left. That is, all that disrupted sleep gets better if you sleep on a rocking bed. So Dave Rappaport, my colleague, who's the head of the sleep research here at the medical school, and I went to New Jersey Institute of Technology, enlisted some senior biomedical education students, and invented a rocking bed. Now, before I show you the rocking bed briefly, I need to tell you that COVID has really disrupted recruitment for this study, and so we need you. If you have CFS and are taking no brain active meds, that means no sleep, no pain, and no antidepressants, please call our research line 212-844-6665. We really, really need you to participate if you live within 50 miles from New York City. So here's the rocking bed. And we compare sleep on this rocking bed to when it's stable. That's the whole research protocol. And then I want to move on to our third funded study, which is, of course, the first in our part of the medical school that's going to compare COVID CFS to classical CFS. And as Dr. Koroshetz said, and as I was worried about, COVID has produced an epidemic of chronic fatigue syndrome. And so the research question is to determine if CFS unassociated with COVID is the same as what we're calling PASC CFS. So we received funding from NINDS, and we is myself and a brilliant biomedical engineer, Zhang Zhu, who is a neuroimager here at Mount Sinai, and this is what we propose to do. We're going to look at 3 sets of patients: PASC CFS, classical CFS with age-matched healthy sedentary controls. We're going to put them in the 3T magnet, and we're going to look at their anatomy. We're going to study perfusion to see if they're differences in blood flow across the brain. And then a whole different step, we're going to look at brain metabolism via venous oxygen extraction. And each of these should provide very important data, the anatomic, whether there are any structural abnormalities, the perfusion, are we going to be able to replicate our prior work showing reduced blood flow? And I'm very excited to see if there are global energy deficits. And each of these, we then will link with the patient's self-report of symptom severity to see what the relation is between these brain dysfunctions and CFS. So the last funded study has to do with a hypothesis that data from 2 consecutive cardiopulmonary exercise tests or stress tests inform us about PEM. So to test that idea, we are doing these 2 CPETs along with something called ecological momentary assessment of symptoms, and I'll explain what that is in a second. And that will allow us to look at the relation between cardiac work and PEM. So we receive funding from NINDS, and I am a co-PI with a brilliant exercise physiologist, Dr. Donna Mancini, a cardiologist here at Mount Sinai. And here's the protocol. Individuals will be sent a wrist-mounted computer which records activity and allows the subject to respond to how he or she is feeling across a number of symptoms several times a day. And they'll do this for 8 days. And then they'll come into the lab where we will determine their blood volume and do the first CPET. Why are we doing blood volume? Because reduced blood volume may be a predictor of PEM. And during the exercise, we use echocardiography, which allows us to assess heart size and heart work, stroke volume. Patient then comes back the next day to do the second CPET. And if the blood volume was reduced on day 1, we randomize the patient to receive a saline infusion or a sham saline infusion. And the reason we're doing this is to determine if increasing blood volume improves cardiac work and reduces PEM. And again, we're using the echocardiography to assess cardiac work, and the patient continues to use the wrist-mounted computer to report activity, and it also records, to record symptoms and symptom severity, and it also records activity, and now we'll be able to track PEM, and then we have the cardiopulmonary and metabolic data to look at the relation between the 2. So now I want to move on to some recent published results that have come from our lab. And the first of these is a collaboration with CDC on orthostatic intolerance in chronic fatigue syndrome. And we did the NASA lean test on 63 CFS patients. And I guess the first most important thing to report to you is that the majority of the tests were abnormal, and the most common was POSH. What's POSH? Well, what POSH is postural orthostatic syndrome of hypocapnia or hyperventilation. We decided to use POSH because it was the same, sort of the same as POTS, postural orthostatic tachycardia syndrome. And here you see POSH in a group of patients during a lean test where we look at end-tidal CO2 via nasal cannula. And what you can see is during the lean, the end-tidal CO2 falls beneath 30, which is indicative of over-breathing. So this is either hypocapnia or hyperventilation, the same thing. So this is POSH. Let's see why I can't get the next slide. So as I said, 32 showed abnormal results out of the 63, and the great majority had POSH, and they have normal respiratory rates. So we infer that they're not breathing quickly, but they're breathing deeply. That's what hyperpnea is. Some had POTS alone, some had POTS and POSH together, and importantly, a subgroup were hyperventilating before exercise at all. And I'll explain why that's important in a second. So the bottom line is the majority of patients in this paper had abnormalities. They were mostly respiratory. So if you were unable to look at their end-tidal CO2 via capnography, you would have missed it. So capnography really is necessary when evaluating a patient for complaints of feeling horrible while standing still. Then the next study looked at PASC patients before and during exercise. So we did cardiopulmonary stress tests on a group of PASC patients who averaged 9 months after their acute COVID infection. And we really found 3 major findings. Many had chronic fatigue syndrome. Many hyperventilated before exercise in the baseline condition, and many had odd-looking abnormal breathing patterns during exercise. Nearly half the patients had CFS, and if we had not excluded for BMIs over 30, it would have been about 56%. Half had hyperventilation during baseline, and a non-overlapping half had what we call dysfunctional breathing. So on the left, this is normal breathing during exercise, and you see it's quite smooth. The patient or subject just breathes more deeper and more across time, but that's not what happens in dysfunctional breathing. Actually, we see a little dysfunctional breathing before the exercise, but then with exercise, we see sighs, deep breaths, less breathing, this odd dysfunctional breathing. Now, these Venn figures show which patients had what. CFS group on the left, no CFS, and these are PASC patients, so no CFS on the right. And the first thing to see is that both groups had the same rate of dysfunctional breathing. This is a small preliminary study. There's a hint of evidence that the CFS patients had more hyperventilation, but as I'm going to show you, we need to do more, and we're embarking on that. So here's our planned research. Question number 1. What happens to hyperventilation and dysfunctional breathing seen in PASC? So what we're doing now is we've just submitted a paperwork to the IRB to allow us to do CPET testing on PASC patients. We expect that will again provide us patients that either hyperventilate or have dysfunctional breathing, and we're going to do sleep studies on them because sleep is a metabolic tranquilizer, if you will. That is, if there's anything going on that's not metabolic, then that will disappear during sleep. And so we're going to use sleep to see what happens to dysfunctional breathing and hyperventilation. And we're also, we'll be able to determine what the differences are between PASC patients that do not have chronic fatigue syndrome and PASC CFS. Our next study that we're just submitting is going to use oxaloacetate supplement to see if it reduces, if it increases the reduced glutathione that we have reported to occur in the cortex of patients with chronic fatigue syndrome. So these patients would be neuroimaged before and after taking 6 weeks of oxaloacetate. And the last study is one that I'm going to do all on my own because I care for PASC patients. And while I can use the same approach I use in ME/CFS, we really need more tools. And I found a paper that vagus nerve stimulation improves the symptoms of PASC. And so we have done prior work with vagus nerve stimulation, but it required implantation, which was a very expensive and surgical procedure. And now we're proposing to be able to stimulate the auricular branch of the vagus, which will allow us to deliver relatively long periods of vagus nerve stimulation to see if that improves PASC. And finally, I need your help for all these studies. So patients with ME/CFS alone or ME/CFS following COVID, you'll get a free workup. You'll be reimbursed for your time. You have to live relatively close to New York to do this. But if you're at all interested, you can get more detail about these studies if you Google Nadelson Lab at Mount Sinai or call our research line 212-844-6900. 6665. Thank you for the honor of letting me speak to you.
Thank you very much, Benjamin. We'll come back with some questions that I see both in the chat and that we've received in advance. But I see that Dr. Nath has joined us, so can we go over to you, Dr. Nath, to give us an update?
Yeah, sure. First of all, let me just say that I enjoyed your presentation, doing a lot of really nice work. Congratulations. So, so as you know, our ME/CFS study had to stop enrollment at the beginning of COVID because we weren't allowed to bring anybody. So then we went ahead and collected a lot of samples. So we started analyzing all those samples. It's best to do it at the end of the study where you don't have variability between assays. So now a lot of the data has been collected and analyzed. We're in the process of writing up our first large manuscript. And so it has a lot of moving parts, but nonetheless, it's coming along well. So our hope is to get it into at least the review process, maybe within the next couple of months or so. And then I think subsequently there will be a number of other manuscripts that will come out on various other parts of the data. So there should be a series of them coming out in a short while. Then just like Dr. Nadelson said, we're doing the exact same thing. That is to look at the past patients and bring them in and compare them with what we've done with the ME/CFS patients and do the exact same testing that we did on them as well. So that way we could compare the two cohorts and see where there's similarities and differences. So that's where we stand at the moment.
Great, thank you very much, Dr. Nath. So we'll move to some questions, and I'll start with some questions for Dr. Nadelson first. So I have a question first about the rocking bed study. If you're involved in that study, is it a one-night study, or is it like over multiple nights that you would have to participate in that study?
Well, it's, Vicki, it's 2 naps, 200-minute naps during the day. Well, what we do in that very briefly is we give patients an actigraph to wear on their wrist for 8 days. So we have some idea from their activity, some information about their sleep. And then before we study them, they wear a home device to make sure they don't have sleep apnea. And once they've been through that introductory part, they come into the sleep lab for a 100-minute nap that has to be separated from the last nap by at least 1 day. So it's every other day. And the other thing is this is a fussy study. If for menstruating women, we can only study them in the first half of their menstrual cycle because the first half and the second half of their menstrual cycle are different in sleep. So if the woman is menstruating, that adds another sort of, not limitation, but requirement. So sometimes we have to study them a week apart, but we try to do the 2 naps within 1 week, Vicki.
Okay, thank you. So a question that came in from the audience is, in your study on PASC-CFS versus CSF, how are PASC-CFS and CFS defined? How are you defining that?
We are going to use the IOM criteria for both groups. I think instead of inventing a new case definition for PASC, I am a big adherent of using published case definitions. So we will identify the group of patients who fulfill the IOM criteria for chronic fatigue, ME/CFS, and then we'll look at the ones that don't. Why don't they fulfill the criteria? What about them? I mean, if they're very similar, well, that's one thing. But if they have very different symptoms, because besides asking the IOM questions, we ask them the questions from way back in 1988, all the symptoms that were initially thought to reflect CFS, and that's 10 symptoms. So we have information about 10 CFS-related symptoms and can use that to block out diagnosis.
Great, thank you. And another question is, in your studies, how will you measure blood volume?
Well, it used to be pretty complicated, but a company invented a device called the Daxor, DAXOR, and that allows us to inject, have the patient come. We inject a tiny bit of radioactive isotope, less radioactivity than a regular chest X-ray, and then we take 4 or 5 blood samples, and the machine tells us their blood volume. It's actually quite wonderful because the old method used to require several isotopes, and it was much more unwieldy.
Thanks. I'll just see if any of the panelists have any questions for Dr. Nadelson.
Hi, this is Walter. Thanks very much. I was wondering what you've seen in terms of post-exertional malaise in the COVID patients.
Plenty of it.
Fatigue, yeah.
Plenty of it. Yeah, I mean, to me, the only difference, Dr. Koroshetz, is that the PASC-CFS, that's what I'm calling them, the PASC-CFS patients have more chest, they have more palpitation, they have more chest pain, they may even have more shortness of breath, although that's hard because classical CFS patients complain of shortness of breath on exertion, but some of that, of course, is due to the fact that their ailment makes them rest and they get deconditioning. But the two ailments overlap, except for those cardiac symptoms. I think those are more common.
Thank you.
Any other questions from the panel? If not, there's a question, why do you use the term CSF and not ME? And I know that there are other questions from people about the terminology for the disease. Dr. Nadelson, do you want to respond to that?
Yeah, well, no, I call it chronic fatigue syndrome because I'm old, and I've been calling that since 1988 when I first started studying this illness. And ME, myalgic encephalomyelitis, is a term that came out of the Iceland mini epidemic many years ago. And those patients indeed did have myalgic encephalomyelitis. They were left with neurologic abnormalities and signs. I do not believe patients with ME have encephalomyelitis, thank goodness. That's why I call it myalgic encephalopathy. They certainly have achy muscles and they certainly have brain dysfunction.
Great, thank you. So we got some questions before, and I think people who submitted questions in advance. And so I think several of the questions really focused on how we are going to be able to, or how will NIH utilize the information coming out of the RECOVER studies to advance research on ME/CFS. So who wants to take that question?
Well, yeah, so this is Walter. You know, as I said in my opening remarks, we've kind of been stymied in ME/CFS because we haven't had, well, thank God, well, until now, we haven't had, you know, thousands of people coming down with the illness all at the same time with a known virus. So the goal we have is to understand the chronic fatigue, ME/CFS that comes from COVID. And as soon as we have clues from that, then to see if those clues generalize to ME/CFS, or at least subgroups of ME/CFS. So I see the RECOVER initiative as, you know, getting at the biological drivers that we've never been able to nail down before. And I think that, you know, this is really an opportunity to understand the fundamental nature of ME/CFS. Now, Dr. Nadelson, am I naive here, or what do you think?
I totally agree, Walter. This When COVID hit, the first thing I did was read about the SARS mini epidemic in Canada, and 25% of those patients fulfilled criteria, 1994 case definition for chronic fatigue syndrome 4 years out. So I was afraid there was going to be an epidemic of CFS, and we are seeing it now. These are people, and we're going to learn from it. This horrible epidemic of CFS is going to do two important things for us. First of all, doctors want to dismiss CFS. They often stigmatize the patient. They did the same thing in fibromyalgia, and then there were 3 FDA-approved drugs for fibromyalgia. And when there were FDA-approved drugs for fibromyalgia, the practicing community changed its mind and started believing in FM. This epidemic of CFS has to make the practitioner understand that this is a post-viral syndrome, and these patients are devastated by this, and they can't be dismissed as, quote, all in your head. That was why I wrote a book about this 12 years ago. It's not all in your head. It may be in your head because it's neurological, but not psychological. So yes, Walter, we really are seeing a lot of it, more than even I was worried about, and Recover should help. And then individual scientists and physicians like myself need to be applying with their own ideas, because Recover is going to give ideas to a group of investigators that are going to sort of come from the top down. And so our scientific progress is always helped when investigators around the water fountain talk and come up with ideas, sit down and write the grant. And that's what I did with Zhang Zhu. She and I talked about chronic fatigue syndrome and PASC, and we put together an application which was just awarded by your institute funding about a month ago, sir. We were grateful. And that's the first that's going to look directly at PASC versus classic CFS.
Yeah, I think also there was a question about stigma. And so, I mean, I hopefully make it absolutely clear that NIH believes that stigma is a horrible thing, particularly for people with ME/CFS. They're suffering enough, and then they have to deal with stigma. But I think that what you're getting at, Benjamin, is that potentially this will decrease the stigma associated with ME/CFS, because people are seeing all the post-COVID patients, and as you said, now that it's right in front of everyone that this is a major public health issue.
Right.
Hopefully that will decrease stigma. But I want to make perfectly clear, NIH is always trying to get rid of stigma for all our diseases.
Well, there's no question that that's the mission, and that's what your funding does, Walter. Your funding brings data, and there's nothing like data to make the point that chronic fatigue syndrome is a brain disease, and we need to understand. That's why you're funding it in NINDS, because it is a brain disease, and we are going to, I am committed to understanding this illness and to helping the patient cope with the disability that's inherent in just receiving the diagnosis.
So I'd like to apologize to Robert Goldsmith. I'm not trying to censor your questions. I'm just simply trying to get through as many questions as we can. So my apologies. And the question, your original question, I assume was what you were referring to that I censored, was, let me go back up here. When will Dr. Koroshetz stop publicly calling the disease chronic fatigue, a symptom, or CFS, and be uniformly respectful and call it ME/CFS or ME?
Yeah, I apologize for going back and forth like that. I should stick with ME/CFS. And we'll do so. Thanks. Good point.
So there's a question from Court. Many new findings are going to come out of RECOVER. How will NIH ensure funding is available to generalize them to ME/CFS? So I first can say that currently under RECOVER, there are funding announcements that have gone out for administrative supplements, and there are ME/CFS investigators who have submitted applications for supplemental funds where they will be able to add cohorts of PASC or long COVID individuals with long COVID to their ongoing ME/CFS studies for very direct comparison utilizing funds from RECOVER, the RECOVER initiative. In addition, in two of the centers currently funded, the Columbia Center under Ian Lipkin, as well as the JAX Center, the PI is Durya Unitmats, we approved pilot projects for both of those centers to be able to collect biospecimens and clinical data from individuals with PASC, again, to be able to very directly compare those results with the results they have from individuals with ME/CFS. In addition, in the new RFAs, individuals are able to, the focus of those RFAs continues to be on ME/CFS. However, if an investigator and their team would like to have a comparator group of individuals with PASC or long COVID, they will be allowed to write that into their grant application. And I'm certain that there will be additional funding announcements coming out of the RECOVER initiative that will be open to individuals, investigators, including investigators who are currently doing research on ME/CFS to do those comparator studies.
I can also see here.
Sure, go ahead.
And yeah, the questions about us showing leadership and repudiating the false narrative that ME/CFS is psychological. Why does seem to be some apprehension to defend this patient population? And I mean, it should be really clear to everybody that NIH is all about trying to understand the biology underneath ME/CFS and never to say that it's all psychological or psychological. So people should just, that's not an issue. NIH is not saying this is psychological. Benjamin, his talk, he mentioned it. I've mentioned it hundreds of times. Let's put it behind us. Let's try and work together to find out what's the cause of the problem and what we can do to treat it. But we're all together. We all agree on this. Absolutely. Same thing with the stigma question. We all agree stigma is horrible for people who are suffering with ME/CFS. I have the same problem with epilepsy. Stigma is horrible for people with epilepsy. It's not. This stigma is a real threat to people's health, and that's our position on this. Okay. Other questions?
Yeah, another question. Do you see a link between EBV, Epstein-Barr virus, and ME/CFS? Benjamin or Dr. Nath?
Let me try. First of all, we heard recently that if you're sick with COVID and you have active EBV, that's a risk factor for COVID, for long COVID. So that's one, but that doesn't tell us anything about EBV in long COVID. There was a paper, I don't believe it's been replicated, having high titers of EBV, early antigen, in survivors, but they were soon after EBV, after acute COVID. We know that titers of EBV are significantly elevated compared to healthy controls, but there is a huge overlap. And in my practice, whenever I suspect it, I look at the one antibody that tells you about reactivation, and that's EBV early antigen. And if it's elevated, I do PCR. And I have to tell you, in 30 years of practice, I personally have not had a positive, but I know my colleague Dan Peterson, who sees many patients, has found positive PCR. And so there are a small number who do, but it's still a research question. What about if you have elevated levels of EBV early antigen? Is that somehow involved in long COVID? We don't know.
Okay, we have time for one more question. Please send any additional.
We're funding a project in Chicago looking at EBV, infectious mono, and chronic fatigue syndrome. The problem with EBV is that it's with you all the time. You never get rid of it. And as Dr. Nadelson said, in acute COVID, you get a reactivation of EBV, but as Dr. Nath taught me, that's true about almost all infectious diseases, so it's not unexpected. But it is a sign from this paper that came out that the higher the EBV titers of our, actually it was the, I guess it was the RNA?
No, I think it may have been VCA, I'm not sure.
Yeah, so anyway, the higher the levels, the higher your chance of getting PASC. And that could be because the higher levels are in the more severe patients. The more severe patients, more likely to get PASC. It's not clear what's cause and effect, I guess.
Yeah, the grant that Dr. Koroshetz is referring to is a grant from NINDS to Leonard Jason, who also received a supplement to add a long COVID or a PASC cohort to that study, looking at individuals who have mono and now have developed, who got COVID, had mono, got COVID, and now have PASC, symptoms of PASC. So the last question, which is a tough one, I think, but I'll ask it anyway, is someone was asking, how does mitochondrial dysfunction fit into the whole picture of, and the question actually, how does mitochondrial dysfunction fit in with CFS research? Anyone want to take that one?
I'll take it, Vicki, because I think that line of research is being addressed in several of the collaborative research centers because of the basically trying to correlate things like post-exertional malaise and overall energy metabolism. And so given the role of mitochondria, I think that's been a natural place to look for clues. Dr. Nath may have done this in his intramural study, but the extramural community is certainly focusing on that because it's a natural place to look with regard to energy metabolism. And I think there are some preliminary results that are intriguing, and I expect we'll see more. And there's been some clues also from work that's now in the published milieu from Norway and other places. So I think it's emerging. Dr. Nath, you have your hand up. Maybe you want to add to that.
So so you're absolutely right. I mean, mitochondria produces energy, so you have low energy, you think, okay, there must be some mitochondrial dysfunction. But mitochondria is very different than other parts of the cell. You know, the mitochondrial abnormalities you see in the muscle, you won't find in the brain. You know, what you find in the brain, you won't find in the heart. So the PBMCs, there can be very different in different parts of the body. That's why many times, if you want to look at mitochondrial DNA and RNA, you have to go to the source. So it's not easy to study. You have to first decide, okay, if this is a muscle problem, then you go after the muscle mitochondria. You think this is a problem in the lymphocytes and lymphocyte function impaired, then you got to look over there. So it should be studied. I'm not sure how easy it's going to be and how easy it's going to be to figure it out. And then the other thing about mitochondria is that a number of the proteins in the mitochondria come from the nucleus. Then there's some that are specific to the mitochondria. Now you got to figure out which one is which.
Yeah. Thank you. Well, thanks to all of our panelists and thanks to our audience for excellent questions. And I'll turn it back to Barbara.
Great, thank you, Dr. Whittemore. A recording and transcript of the call will be posted to the NIH MECFS website soon. In closing today's call, I'd like to remind you about our listserv for updates from NIH. To be added to the listserv, please visit the NIH MECFS website at www.nih.gov/MECFS and click on Join Our Listserv at the bottom of the left sidebar. Thank you for an informative and thoughtful discussion, and have a great rest of your day.